Pediatrics in Imperial College London, my academic interest has been in the early life origins, particularly of allergic and respiratory disease. And this area has become of enormous interest worldwide in trying to understand the causes and consequences of early life events and how they impact on later susceptibility to disease. So for my first slide, I'm showing an ancient picture of the Madonna and Child, and clearly the perspective of the child as a miniature adult is something that's changed spectacularly over the last century. And I do rather like this quote from Simone de Beauvoir, who said, "What is an adult? Basically a child blown up by age." William James Mayo and his brother, who set up the Mayo Clinic, has a wonderful quote which I think underlies the whole idea of early life origins of health and disease, and that is that the ideal of medicine is to eliminate the need of a physician. And as far as I'm concerned, if one could understand the early life origins of disease, then we will achieve the ideal. This all, in terms of modern perspectives, arises from David Barker and his research, which led to the Barker hypothesis, which is that the seeds of most diseases are sown during organogenesis and periods of rapid cell division, when even minor perturbations can have a lifelong impact that is far greater than any perturbation that occurs after organs are fully formed and cell division is less rapid. So I'm going to take you through observational studies, often from epidemiology or clinical practice, and then think about the ways in which they can be pulled together in triangulation to generate a hypothesis which might explain the associations which have come from the observational studies. Clearly that's then followed by trying to understand mechanisms that may be involved, which in turn will identify targets for intervention, and then the final proof of the pudding will be in intervention trials to see whether they truly will prevent the evolution of disease. Some of the best examples of observation of the early life origins of disease come from looking at migrating populations moving from one environment to another. My example here is of asthma prevalence in Chinese adolescents living either in Canada or in China. So one is assuming here that the genetics are the same, and the incidence of asthma, or the prevalence of asthma, I should say, in adolescents living in China is 2%. If they have migrated to Canada and have only been there for less than 7 years, that prevalence rate goes up to 7%. If they've been in Canada for more than 7 years, it becomes 12%. And most importantly, if they're born in Canada, in other words, they're the second generation immigrants from China to Canada, then the prevalence of asthma becomes 16% and is very close to that of the native Canadian population. This is pointing to there being some very important early life events, but also that there are events occurring later on if there's a constant exposure to a new environment. What factors may be involved? Well, if we're thinking about pregnancy and being born in Canada, one issue may be an effect on nutrition and therefore the growth of the fetus. And David Barker in his original work showed a correlation between coronary artery disease in mid to late adult life and birth weight. So this is a figure of one of his original papers looking at the standardized mortality rate from coronary artery disease in relation to the birth weight of the individuals involved. And as you will see, there is a far higher mortality rate for those born less than 5.5 pounds compared with being of medium or high birth weight, although having an excessively high birth weight above 9.5 pounds is perhaps associated again with a slight increase in mortality rate, and clearly that is going back into an abnormal range, with perhaps many of these babies having been born to diabetic mothers, which will have had a considerable impact on subsequent susceptibility to disease. This has been shown for many other organs and susceptibility to disease in many other organs. And another component of his research was to look at lung function in individuals in mid to late adult life from 59 to 70 years of age, and this is the forced expiratory volume in 1 second, again related to birth weight. And this is taking into account subsequent weight and height and making allowances for other confounding factors, and still showing that being born with a low birth weight below 5.5 pounds is associated with a lower lung function in late adult life, with a very big difference and a consistent gradient of increasing lung function with increasing birth weight. We can think about the way in which lung function evolves by looking at how it develops through life from birth onwards. A newborn baby, in terms of its overall lung function, this is taking into account many factors, is about 70% in terms of efficiency compared with a young adult. Over the first few years of life, there is a rapid increase in new alveolus formation and in setting up of all the interstitial tissues and improving elastic recoil of the lungs, so that by 2 to 3 years of age, they're getting close to the perfect 100% efficient lungs. And by early adulthood, they've reached 100%. Sadly, as you age, you lose elastic recoil within your lungs, and lung function will naturally diminish, and the late adult life is perhaps equivalent to the newborn baby in terms of having returned to about 70% efficient lungs. Now, what happens if you're low birth weight? And perhaps one of the most obvious causes of low birth weight is if the mother smokes. Well, again, the lung function will progressively improve with growth of alveoli, with enlargement of the airways, with improving transpulmonary pressure, with the recoil forces exerted by the thoracic cage improving as the ribs ossify. So the lung function will improve, but it will remain somewhat subnormal, maybe only 80-85% of normal. That will be associated with absolutely normal activity and no evidence of any problem in a young adult because there's a large respiratory reserve. However, again, as elastic recoil diminishes with aging, so lung function will fall away, and it will be then significantly below 70% in the elderly adult, and may only be 50% of what it should be, and then there can well be a few problems. And if you superimpose postnatal insults, which might occur sequentially through childhood, then the lung function, even in the young adult, is now submaximal. And as it falls away in the elderly adult, you will end up with severe and significant respiratory problems, such as chronic obstructive pulmonary disease. And that condition must be seen as an evolution of problems which has occurred through conception, childhood, and adulthood, heaping on the insults to cause progressively more problems with lung function. The concept of the early life origins of disease is based on a concept of so-called fetal programming, and that is that as the fetus is growing, factors in utero as they're growing up, programming the way not only the fetus will continue to grow, but the way in which the child postnatally will grow. And this impacts on susceptibility to cardiovascular disease, type 2 diabetes, lung disease, as I've indicated, even cancer risk, neurological and intellectual impairment, and allergic and autoimmune diseases. There is a hypothesis that's arisen, known as the evolutionary hypothesis, which suggests that as you grow and develop in utero as a fetus, you are programmed as a consequence of the environmental factors to which your mother is exposed during the pregnancy to be able to handle that environment into which you're going to be born. And the programming involves epigenetic changes, effects on gene transcription, altered tissue differentiation and altered homeostatic processes. And provided there is a concordance between the intrauterine environment, in other words, the environment of your mother during pregnancy, and the environment into which you're born, you will be equipped to deal with that environment. So your extrauterine life will be dictated by genotype and phenotype at birth. But if there's a mismatch between what you're going to be exposed to after birth compared with what your mother was exposed to during pregnancy, there will be differences in endocrine, metabolic and immune responses which will make you more susceptible to problems postnatally. So if we look at this a little further, fetal gene expression is going to be influenced by maternal health and environment, and if that is in some way aberrant, it could affect fetal nutrition and thereby result in the fetal nutrient demand exceeding supply. That in turn will affect the endocrine responses, and there will be impacts on placental vascular supply. As a consequence, the fetus will have altered endocrine and metabolic processes, will have differences in bone, lean and fat body mass, will have altered blood flow, vascular loading, and altered immune responses. And then postnatally, there will be amplification of these problems if there are other exposures that impact on these variations, leading to cardiovascular, metabolic, lung and immune-mediated diseases. So let's take one example, maternal pregnancy smoking. We know that this will cause intrauterine growth retardation. It increases a whole host of pregnancy complications. It increases the risk of the infant having sudden infant death syndrome, of respiratory illnesses in infancy, and there are also data suggesting poorer educational attainments. As far as the impacts on the lungs are concerned, there are a lot of data now showing that smoking has a profound effect on the way the airways grow and develop. So normally the infant is born with about 50 to 60% of the adult alveolar number and with a particular airway caliber. Through early infancy, over the first few years of life, there are new alveoli forming, and of course the airways are enlarging, so that by early adulthood you have a nice big airway with lots of alveolar tissue support to enhance elastic recoil to hold the airways open even as the intra-airway pressure falls during expiration. As you go into our old age, the elastic tissue is no longer as good, and so elastic recoil forces diminish. But because the airways are of reasonable size and there are a lot of alveoli around the airways, the caliber of the airways, even during expiration, remains reasonable. The infant of the smoking mother starts off with a smaller caliber airway. This in part because smoking is impairing fetal respiratory movements, which are important for having a trophic effect in allowing the airways to grow. But there's also a diminution in the number of alveoli. During childhood, the alveolar number will increase, but it'll never reach that of the healthy adult born to a non-smoking mother, and there will be more alveolar septa to support the airway, but fewer than there would otherwise have been without the smoke exposure. And then when we get to the old age, the elastic recoil forces have gone, you've got a smaller airway with less support, and there chronic obstructive pulmonary disease has evolved. The next thing we must consider is how do genes and environment interact? We now have the full map of the human genome, and whilst our expectation was that it would explain the majority of disease, clearly that has not been the case, and that actually genome explains relatively little human variation. So now let's understand the environmental factors affecting the genes. It could be that the genetic impairment, say, of immune integrity or of metabolic processes, together with exposure to a particular stimulus, in the case of immune development, if it's impaired, if we expose ourselves to an inflammatory stimulus, then there's going to be a greater problem. It could be that the genetic impairment of a metabolic process and then exposure to the metabolite will cause the problem, as in phenylketonuria, for example. But the new kid on the block, as it were, the new area, which is a very rapidly expanding and exciting area, is in epigenetics. So let's look at a few examples. Here's a risk of asthma associated with a genetic defect in an antioxidant gene, a glutathione methyltransferase gene, which is very important in conferring antioxidant defenses, and how that impacts on environmental tobacco smoke exposure and thereby changes susceptibility to asthma. So if one has a normal gene and no environmental tobacco smoke exposure, the risk of asthma is lower than the odds ratio for the whole population. If you're exposed to tobacco smoke, but the gene is normal, then again that does not increase your risk of asthma, and the increased risk of asthma is purely in those who have both the genetic defect and the exposure to tobacco smoke. And if it's in terms of having more severe asthma that ends up in the emergency room, in accident and emergency, then the amplification of risk is very considerable, and at 3.5 times or more the risk of either having a normal gene or not being exposed to tobacco smoke, or both. And that's similar for other environmental pollutants. This is a very specific antioxidant gene polymorphism in glutathione S transferase P1 gene in individuals who are either in a district with low air pollution, moderate air pollution, or high air pollution. And it is only in the high air pollution area that this genetic defect shows itself in increasing the odds ratio for developing asthma. And interestingly, these polymorphisms are also associated with susceptibility to developing asthma if there is a significant exposure to paracetamol, very commonly used as an antipyretic, analgesic in young children. It's the first thing mothers turn to when their child is irritable and unwell. Paracetamol has a metabolite which has to be removed by antioxidant defenses. And if there are polymorphisms in the antioxidant gene, then this metabolite is not removed and can then contribute to increasing the risk of developing asthma. And so this is the prevalence of wheeze in 5-year-olds, dependent on whether they have days of being given paracetamol, and as this is an American study, it's labeled acetaminophen, and having a lot of acetaminophen and the genetic polymorphism increases the risk of having wheezing at the age of 5 very considerably, with more than a doubling of the risk with the combination of being given the paracetamol and having the gene polymorphism. So let's look at the way in which epigenetics might contribute to understanding of these interactions. And perhaps the simplest example of epigenetics is to ask the question, what makes a queen bee, nature or nurture? A queen bee is morphologically and functionally very different to the workers, but has absolutely identical genome. And the reason that the queen bee is so different is because of the epigenetic effects of royal jelly. In other words, the food that the larva is fed on during its early development, which changes it morphologically, changes its phenotype very dramatically in the presence of the same genotype. So epigenetics is defined as changes in the phenotype or gene expression caused by mechanisms other than changes in the DNA sequence, which may be transmissible across generations. And if we go back in time, actually this supports the concept of Lamarckism. Lamarck actually suggested that there were acquired heritable characteristics, which many people nonsensed at the time, and with the onset of Darwinism, Lamarckism was lost, and now it comes back in the form of epigenetics. There are a number of mechanisms that are involved that are now being disentangled to understand how environmental factors can affect gene expression without modifying DNA sequence. One of them is that some components of DNA can be methylated, and those are the CpG sequences. It's particularly the cytosine in the CpG sequences that becomes methylated. And the methylation then changes gene expression. It actually impairs gene expression. The other thing is that the DNA is wound round molecules known as histones, and the histones have tails which can also be chemically modified by methylation, acetylation, phosphorylation and ubiquitination, which will again change the way in which the DNA sequence is going to be translated, this, in this case, often the histone modification opens up the DNA sequences, so it's more easy for them to be translated. So histone modifications increases gene expression, DNA methylation decreases gene expression. A nice animal model of this is the agouti mouse, which is normally obese and prone to diabetes and cancer. But if the mother during the pregnancy is fed with folic acid, which is a methyl donor and will therefore affect the histones and the DNA, then you can change this obese diabetic mouse into a very normal, different coloured mouse that has a very different outcome. So the mechanisms, as I've said, are DNA methylation to CpG motifs, and there are 30 million in the human genome, which impairs transcription, or histone modification on 30 million nucleosomes by these chemical modifications, which opens the chromatin to aid transcription. There is a third mechanism by which gene expression can be affected, and that is simultaneously with messenger RNA being generated to move into the cytoplasm of the cell and lead to the production of relevant proteins. There are microRNAs also released into the cytoplasm, which will block the messenger RNA and therefore block the full transcription process. So that's the mechanism. So let's look at some other examples of dramatic perturbations that are occurring during pregnancy, which have an impact throughout life. So one of the best examples is the so-called Dutch famine study. So towards the end of the war, and in the first months after the end of the Second World War, the Dutch population was exposed to a pretty drastic famine. So it's very tightly timed, and it has been possible to look at children who during pregnancy were exposed to the Dutch famine. So there are children who were born before the famine, there were children who were born with exposure to the famine only in the last trimester of pregnancy, some exposed in the mid-trimester and some exposed very early. So if conception occurred around the time of the onset of the famine or shortly before, then they will have been exposed to serious poor nutrition early on in pregnancy. The individuals exposed to the famine during pregnancy have been followed for throughout life, and there are data now showing that dependent on when the famine occurred, there are different susceptibilities. So those who had nutrient impaired delivery early in pregnancy are more prone to coronary artery disease, have raised lipid profiles, abnormal clotting, and paradoxically are more prone to obesity. If the exposure was in mid-pregnancy, they're more likely to have obstructive airways disease. This is at a time when the airways are growing and developing, so not surprising that this is the case, and also to microalbuminuria. Exposure in late pregnancy was associated with impaired glucose tolerance. More recently, people have looked at the degree of methylation of particular genes in relation to exposure to the Dutch famine. And this is slightly complex. This is comparing people who were exposed during pregnancy to the Dutch famine compared with unexposed siblings, or who are the closest match one can have, looking at the amount of methylation of the gene for insulin growth factor 2. And there is hypomethylation, reduced methylation, if the exposure to the famine occurred during pregnancy, but if the famine exposure was after birth, then it has had no effect. So here is this very early influence during the pregnancy affecting methylation and thereby changing expression of a particular gene. What isn't clear here from this study is whether the methylation is on the DNA or on the nucleosomes, on the histones. There are other studies now looking at epigenetic effects on susceptibility to autoimmune disease. And while there are clearly genetic polymorphisms, effects of gender and mutations to genes which will have an effect on susceptibility to autoimmunity, there are a whole range of environmental factors, environmental exposures, drugs, viruses, diet and nutrition, which could be impacting on genes. And so it's been shown that there's a low concordance in monozygotic twins for autoimmunity. But if you look at individuals with autoimmune disease, such as systemic lupus erythematosus, there's global hypomethylation of genes, meaning particularly in relation to immune response genes and to Th1 responsive genes, that there will be increased expression. Rheumatoid arthritis it's been shown that the CD4, that's the helper T cells in joints, are demethylated in relation to interferon gamma. And interferon gamma is an archetypal Th1 cytokine which has a major impact in relation to susceptibility to autoimmune disease. There's also hypomethylation of the FOXP3 gene, which is the transcription factor within regulatory T cells. Primary biliary cirrhosis, another autoimmune problem, is associated with demethylation of CD40 ligand. That's the cluster of differentiation 40 ligand, which is expressed on lymphocytes and is very important in terms of signaling between T cells and B cells. Folic acid, as I indicated before, is a methyl donor, as has been shown in the agouti mouse. We now give folic acid preconceptually and in early pregnancy to prevent spina bifida, and it may well be doing so by modifying gene expression. But actually it's more recently been shown that if the folic acid is continued throughout pregnancy, there's an increased risk of those infants subsequently having wheezing or lower respiratory infections or admissions for lower respiratory infections. And it is assumed here that the folic acid is, again, as a methyl donor, modifying gene expression, and whilst it might be reducing spina bifida, is changing immune responses and making individuals more susceptible to airway inflammation and therefore to wheezing. I've mentioned microRNAs as inhibitors of messenger RNA and therefore of transcription into proteins. There's enormous heterogeneity, so one microRNA can affect many different messenger RNAs, but there are over 1,000 human microRNAs that have been identified, and many of them, their target sites have now been identified. Thinking about how viruses might have an epigenetic effect, well, RSV, respiratory syncytial virus, has RNA sequences which have high sequence homology to human microRNAs, and it may well be that RSV has its impact on immune responses by impairing transcription of messenger RNAs, which are important for eliminating the virus, so it's defending itself. But this can have a fairly prolonged impact, so we know that children who have had RSV bronchiolitis can have recurrent wheezing for many years after the initial infection, and this could well be the microRNAs sticking around and having a more lasting effect on responses to subsequent infections. Viral-induced dysregulation of microRNA may induce changes in innate immune responses and in Th1, Th2 and T regulatory cell balance. Th1 associated with protection against infection, autoimmune disease and organ rejection in transplant. Th2 being associated with elimination of parasites, but also with allergic disease, and regulatory T cells obviously controlling both. It's very interesting. There are high levels of microRNA in human breast milk. The question of what they're doing early on in infancy is clearly something that requires a lot more research. I've mentioned insulin growth factor 2, which is raised in obese individuals. People who are obese have excessive methylation of the IGF-2 gene. This is a study that was done in 1-year-old infants who subsequently became obese. This seems rather paradoxical because I've said methylation of the gene will reduce expression, but actually paradoxically in this case it's associated with increased transcription, so maybe the methylation is more on the histones than on the gene itself. And as I pointed out, if microRNAs in breast milk have an impact, well, is there an example that this is the case? Here we are. There is the effect of any variations in methylation of the IGF-2 can be offset if the individual has been breastfed. Is an epigenetic change transmissible? Well, perhaps the best example is going back to cigarette smoking. There is a remarkable study which has shown that while you have an increased risk of asthma if your mother smokes during your pregnancy, remarkably, if your grandmother smoked during the pregnancy of your mother, even if she doesn't smoke during your pregnancy, you still have an increased risk of having asthma. And if you put the two together, which is most commonly the case, you have 2.5 times the risk of developing asthma. So make sure your mother and your grandmother don't smoke. I now want to move on to thinking a bit more about immunology. There's a wonderful quote from Miroslav Holub, who was an immunologist but also a poet, a Czech poet. He wrote a book called Zen and the Thymus, and in this he said, immunologists are the function of a culture in which jumping to conclusion means endangering survival of men, women and infants. In other words, if you don't understand everything and you make judgments and do something about it without a full understanding, you could do more harm than good. Well, let's understand a bit about the way in which the immune system evolves during pregnancy. And this will mostly focus on susceptibility to allergic disease, which is my main research interest. This is a study looking at birth weight in relation to susceptibility to having a positive allergy skin test at 16 years of age. And a high birth weight here is associated with a significantly higher risk. This is the odds ratio and the 95% confidence intervals for those born with a birth weight above 4.5 kilos, compared with a moderately increased birth weight, where there's still an increased risk of having a positive skin test, compared with having a normal birth weight between 2.5 and 3.4 kilos. And here's another study, just to triangulate it, which has shown that increasing birth weight is associated with a higher likelihood in terms of percentage of individuals to have a positive IgE antibody of greater than 0.7 international units per mil, going from intrauterine growth retarded to very high birth weight individuals. There is a progressive increase. It is the same for birth length, although head circumference, this is not significant. I'm afraid the p-value here has got shifted on this slide, so the p0.14 relates to the head circumference gradation, which is not significantly different, but birth weight and length are. So what's going on in pregnancy that's influencing this? Well, the fetus expresses paternal antigens as well as maternal antigens, and therefore the mother's immune system should reject the fetus in the same way that it would reject a transplant. And actually maternal immune attack will occur against the decidual tissues. So if this happens, as it sometimes does, there is often early miscarriage. In murine models, it's been shown to cause intrauterine growth retardation, and may do so in human situations as well. So this can happen, but in the majority it doesn't. So we need to understand immune balance here. And it is a bit of a balancing act. The Th1, the T helper 1 cell response, involved with generation of interferon gamma and interleukin 2 under the influence of interleukin 12, is important for eliminating microorganisms and tumours. And if this is impaired, you're more prone to cancer, you're more prone to infection. But if it's excessive and inappropriate in response, it will be associated with autoimmune disease, and of course it could be associated with transplant rejection or recurrent miscarriage. Balancing this response is the Th2 response associated with interleukins 4, 5 and 13, which of course are the archetypal cytokines associated with allergic disease, but they're also important for eliminating parasites. But interestingly, they have considerable importance in sustaining pregnancy, because interleukin 4 suppresses Th1 activity and interferon gamma suppresses Th2 activity. That accounts for the balance. Balancing both of these are the regulatory T cells, which generate interleukin 10 and transforming growth factor beta. What has become very apparent in both human and murine models is that the decidual tissues generate these Th2 cytokines. So this is a stain of human amnion, and this red smudge and the base of the cells is interleukin 4. Vast amounts of interleukin 4 being generated at the feto-maternal interface, which will downregulate the maternal Th1 response. This has a stain for interleukin 13, another Th2 cytokine in the human placenta. And again, the red cells, lots and lots of interleukin 13, having a similar impact. The other thing that's found in the decidual tissues and in the amniotic fluid is interleukin 10 and transforming growth factor beta. These are regulatory cytokines. So the combination of increased Th2 and regulatory cytokines means that the maternal fetal rejecting Th1 response is downregulated and balanced, and therefore there is not fetal rejection. So this is very important to sustain the pregnancy. The Th2 response is balanced because there's both regulation as well as Th2 promotion, although some women during pregnancy do have a deterioration in their allergic disease. And many women with autoimmune disease, interestingly, have an improvement during pregnancy. So this balance can be easily understood. But the combination of this is resulting in the fetus being protected, not rejected. The amniotic fluid contains vast quantities of these regulatory and Th2 cytokines, all of which are swallowed by the fetus. And while the fetal circulation is very immature immunologically, the fetal gut is very mature. And this is a stain for T cells, CD3 positive T cells, in a 16-week-old fetal, a 16-week gestation fetal small bowel. You see loads of T cells. And it's become clear that those T cells are highly mature, possibly under the influence of the cytokine swallowed from the amniotic fluid. But the key issue is that these T cells are capable of responding, and it's been possible to show that there are mature antigen-presenting cells that are communicating with T cells in the fetal bowel from as early as 14 to 16 weeks gestation. So the fetus is swallowing cytokines which are Th2 biased, IL-4 and IL-13. There's increased regulatory cytokines. What the fetus is also swallowing is small quantities of allergen, of proteins that the mother has eaten or been exposed to in her environment. Small amounts get across the placenta, across the amnion into the amniotic fluid, and also maternal IgE. Whilst it does not get transported across the placenta into the fetal circulation, it's present in the amniotic fluid. All of this is swallowed by the fetus, which will present the fetal immune cells in the gut with a Th2 biased stimulus and allergen to push the response towards a sort of allergically biased immune response. And in fact, all newborn babies have a slightly allergically biased immune response, which maybe sets the scene for them to develop an itchy rash, eczema, wheezing, asthma, and sneezing, allergic rhinitis. So how do we link that with the fetus growing rapidly? Well, the Th2 biased intrauterine environment not only protects the pregnancy against the maternal Th1 response, it also enhances fetal growth and programs a rapid growth trajectory during early pregnancy. And if that results in a large neonate, that neonate is at increased risk of being allergically sensitized. But that is only the first step towards allergic disease. In order to have allergic disease, something else must happen. So if it's going to be asthma, there must be something about an effect on lung function. And I suggested to you that faltering growth in the mid-trimester of pregnancy may be associated with reduced lung function. So if in combination with programming for rapid growth and a Th2 biased response to develop allergy, there is some faltering of growth in the mid-trimester of pregnancy, then lung function will not be as good with this growth faltering, leading to the combination of differences in lung function and allergic inflammation, which is the combination leading to allergic asthma. What about extrauterine factors and how they impact? Well, the important one to think about is the hygiene hypothesis. This was perhaps not as new as one might think. Charles Harrison Blackley, in his book which he published in 1873, Experimental Researches on the Causes and Nature of Catarrhus aestivus, which is spring catarrh, now known as hay fever, it would seem that hay fever has of late been considerably on the increase. The persons most subject to the action of pollen belong to a class which furnishes the fewest cases of the disorder, namely the farming class. It has been suggested that if you're exposed like this young child to lots of dirt and animals, that you're going to have less allergy. And the suggestion from the hygiene hypothesis is this is an interaction between exposure to organisms leading to infection, promoting a Th1 response and reducing Th2 responses, and therefore allergy. So if you look at demographic trends in the Western world to infections, hepatitis, tuberculosis, mumps, measles, then those have gone down spectacularly, particularly in the second half of the 20th century. At the same time, as immune-mediated inflammatory diseases like multiple sclerosis, Crohn's disease, type 1 diabetes and asthma have all spectacularly increased. Is this cause and effect? Less infection, more immune-mediated chronic disease? Well, there is lots of evidence to support this. If we look at the prevalence of wheezing by age in children who have either got an older sibling or with under 6 months of age have entered a daycare environment, and then they are exposed to lots more bugs early on in life. And actually early on in life, therefore, if those organisms are affecting the respiratory tract, they're more likely to wheeze, compared with infants who don't go into daycare early or don't have an older sibling. However, as they get older, remarkably, those that had more problems as infants because of lots more exposure to minor infections, subsequently beyond the age of 5, have significantly less asthma and actually less allergic asthma particularly than those who had a nice sterile or relatively less infection-loaded environment, who end up with more allergic disease. And if we try and eliminate bugs by giving lots of antibiotics, both postnatally but even antenatally, the more antibiotic courses that a mother has received during pregnancy, the higher the probability that her offspring will later have asthma, and particularly allergic asthma. Our assumption here is that the antibiotics in pregnancy are modifying the mother's gut microbiome, and therefore that changes the inoculum that the newborn baby receives during normal delivery. This has led to the idea that perhaps we should be rolling our children in the dirt, and this just happens to be a cartoon from an American newspaper picking up on this hygiene hypothesis. It is perhaps most apparent from farming studies, which goes back to Charles Harrison Blackley's statement. In Bavaria particularly, families live on top of their animals, so the animals are in the stables on the ground floor and the family lives on the first and second floor. And so the mother in pregnancy and the baby is exposed to cows and lots of dirt, and actually also often the mother is drinking unpasteurized milk, and once the baby is weaned, the baby goes on to unpasteurized milk as well. And this is associated with a protection against allergy. And this is a compilation of studies from Bavaria looking at exposure to farm environment in the first year of life. If there isn't any exposure, the subsequent prevalence of asthma in rural populations was 12%. In those living on a farm, it was halved to around 6%. If it was related to the family drinking a lot of unpasteurized milk, then that was associated with a lower risk. Being in the stables a lot of the time, similarly, and if you put the two together, the rates of asthma are down to less than 2%, a sixth of what they are outside the farming environment. This has been elaborated on in the UK with a study which has tried to discriminate between exposure to unpasteurized milk and exposure to other factors on the farm. And it would appear that the major effect is the unpasteurized milk. So comparing rural families where there is no farm contact with families that live in villages where the parents work on the farms, but during pregnancy there's no exposure, or postnatally early in life no exposure to the farm, compared with living on the farm, you will see the key effect is if they consume unpasteurized milk rather than where they live and work. If that is the case, then we need to now begin to look at unpasteurized milk and consider what it is in unpasteurized milk that's conferring protection. And this comes back to the gut flora, because it's also become apparent that if you look at the feces of small babies and look at the number of different organisms that are beginning to colonize the newborn baby's gut in the first weeks of life, and then follow those babies over the first year or two of life, those that subsequently become allergic have fewer bugs in their gut early on than those who are non-allergic. So there's something, and it's now become clear that the range of organisms is also fundamentally different. What we look at to enhance normal gut function and normal growth and reduce the risks of all sorts of diseases is to breastfeed our children. And in human breast milk, there's obviously sugars and fat and proteins, but the third most prevalent set of substances in breast milk are oligosaccharides. These are non-digestible sugars which are used by organisms in the gut to facilitate their growth and development. In other words, oligosaccharides are prebiotics. They encourage the development of a normal gut flora. And there have now been studies which have looked at giving babies who aren't breastfed a milk formula which contains prebiotic oligosaccharides similar to those in breast milk. So this is a study comparing the administration of a hydrolyzed whey formula either with galacto and fructo oligosaccharides added to the formula or no prebiotics added to the formula, and then looking at the cumulative incidence of eczema at 6 months of age, and it's very considerably lower in those that were fed the prebiotic enhanced formula compared with those that were just given the hydrolyzed whey formula. So this prebiotic is changing the gut flora and thereby reducing the risk of this early atopic illness. So this is re-representing that same data in terms of the incidence of atopic eczema, atopic dermatitis being less than half in the those fed the prebiotic, simultaneously with there being a difference in the number of bifidobacteria that are in the gut, and it is perceived that bifidobacteria are particularly important in relation to modulating immune responses and being associated with less allergy, and these are the organisms that you would see much more frequently in breastfed babies as well. So in a way this formula was mimicking to some extent one of the breast milk effects, and again suggesting that this is particularly important in relation to moderating immune responses early in life. One might ask the question whether unpasteurized milk is either giving more bugs, which of course it is, with many more lactobacilli in unpasteurized milk, or is it that the unpasteurized milk has more active oligosaccharides compared with modified milk formulae, where the oligosaccharides will be modified by the processing. So I hope you have been convinced that genetics and epigenetics are going to be important in influencing current health. Particularly epigenetic mechanisms may well be impacted on by maternal health, nutrition, and environmental exposures during pregnancy. Obviously, if there are complications during pregnancy impairing nutrient delivery across the placenta, or factors during delivery, are going to have an influence on postnatal health. The way in which the fetus is programmed to handle the extrauterine environment will also impact, and clearly there are early life events that are also impinging. So I'll leave you with a poem from the Lakeland poet from the UK, William Wordsworth, who wrote, "My heart leaps up when I behold a rainbow in the sky. So was it when my life began, so is it now I am a man. So be it when I shall grow old or let me die. The child is father of the man." Perhaps I should add, perhaps the fetus is the father of the child. So is it already too late when the cord has been cut? Lots more research to do. I've given you some recommended reading there. The medical literature is now full of fetal and early life origins of health and disease, or developmental origins of health and disease.